talk from Dr. Michelle Monje, who will be online. So. Oh, here she is. Hi, Michelle.
Hi.
Nice to see you. And I'm happy to introduce Michelle Monje from Stanford University, where she is a professor of pediatric neuro-oncology there. And she has done a pioneering work in the characterization of the connectivity between tumor cells and the normal brain. We are very excited to hear your talk. Thank you, Michelle.
Wonderful. It's so nice to join you. Thank you for letting me join you from my office. I'm going to talk a little bit about the neuroscience of brain cancer. I'm a pediatric neuro-oncologist, and as I think many people in the audience likely know, cancers of the central nervous system in children happen in particular locations at particular ages. And this is especially true of gliomas. In fact, I remember as a trainee in the Partners Program, or what used to be called the Partners Program, now MGB, an attending saying to me, if you tell me where a child's tumor is, I'll tell you their age. And I remember being just really struck by that, and started paying attention in adult glioblastoma to the location predilection kind of overlapping with regions of the nervous system that we know to be relatively plastic and adaptable. And so together, this suggested to me that gliomas in particular, but perhaps brain cancers in general, may be diseases of dysregulated both neurodevelopment and also neural plasticity. And just to introduce the audience to some of the tumors that I'm going to focus on in this talk, this is a low-grade NF1-associated optic pathway glioma. This is a tumor that tends to occur in fairly young children, and I'm showing you an MRI scan here from a 4-year-old. It's a low-grade tumor, doesn't tend to transform. It is, a survivable malignancy, but often causes really severe disability and often blindness. In contrast to the low-grade nature of this tumor, here you see a diffuse intrinsic pontine glioma, also called an H3K27M altered diffuse midline glioma of the pons. This is one of the most aggressive imaginable cancers with a median overall survival of only 11 months. And a molecularly related disease that happens in slightly older children, DIPG tends to happen in mid-childhood around ages 6 to 7. In slightly older children, peaking in incidence around age 10, H3K27M diffuse midline gliomas of the thalamus tend to occur. And again, very diffusely infiltrative. There is no central mass, unlike GBM. There is just this diffusely infiltrative component that affects the entirety of the structure, in this case, the brainstem, in this case, the thalamus, often both thalami. And then in adolescence and young adulthood, hemispheric high-grade gliomas, including kind of the pediatric version of GBM, tend to occur. And when you take a step back and think about, the developmental processes that might correlate with the time and place incidence of pediatric gliomas, it strikes you that developmental myelination is happening in the times and place that these tumors tend to form. And as I think probably many in this audience know, myelin, fascinatingly, develops chiefly postnatally in a process that spans about 30 years of neural development. So, from just before birth until late 20s, early 30s, myelin continues to develop, and it develops in predictable chronological and topographical patterns, such that at a time when there is a discrete wave of developmental myelination in the brainstem, and more particularly in the ventral pons and the corticospinal tracts, this is when diffuse intrinsic pontine glioma tends to occur in mid-childhood between ages of 6 and 7. Similarly, at a time when there's a discrete wave of neocortical and cortical projection fiber myelination, this is when hemispheric high-grade gliomas of childhood, including pediatric glioblastoma, tend to occur. And these observations are concordant with findings from my laboratory and from many others that the origin of these high-grade gliomas in children, but also in adults, is frequently cells that are in the oligodendroglial lineage, in the myelin-forming cell lineage, either bona fide oligodendrocyte precursors, or OPCs, or just earlier neural precursors that are committed to go down this oligodendroglial differentiation pathway. And so we may learn really important lessons about gliomagenesis by better understanding what normally regulates the behavior of these healthy glial precursor cells. And so when I started my lab now about 15 years ago, one of the first questions we wanted to ask was, what does regulate the proliferation and functional differentiation of oligodendroglial lineage cells? And one idea that was in the literature at the time and that I found really intriguing was the idea that neurons themselves may regulate the extent to which their axons are myelinated, and that they may do this in an activity and experience dependent way, thereby kind of tuning the dynamics of a neural circuit by modulating the myelin and therefore the speed of action potential conductance. So we wanted to understand if neuronal activity promotes, changes in the oligodendroglial lineage, particularly OPC proliferation, and whether, in general, myelin might be plastic and adaptable to experience an activity. And what we found was that, indeed, in the healthy nervous system, particular neural circuits, particular neurons exhibit this very clear plasticity of myelin. There are fine-tuned changes that happen in the oligodendroglial lineage, initially with proliferation of OPCs in an activity-dependent way through activity-dependent paracrine factor secretion. Those proliferating OPCs then generate new oligodendrocytes which ensheath regions of axons or entirely unmyelinated axons. And that change in myelin alters tunes the dynamics of a circuit in a way that in the healthy nervous system promotes coordinated circuit function and contributes to learning and memory and other brain functions. So then the question was, if neuronal activity promotes the proliferation of these healthy oligodendroglial cells, what happens if there's an oncogenic mutation in that OPC? Could this really powerful activity-induced regulation of the normal precursor be hijacked and subverted in the context of glial cancers? And so now, about 10 years ago, we leveraged tools of modern neuroscience like in vivo optogenetics to ask, if we modulate the activity of a neural circuit of a particular genetically encoded subtype of neuron, does that influence a glioma that may be growing within that circuit? So we performed optogenetics stimulating initially these cortical projection neurons in the motor planning area. This evokes some complex motor output from the mice, so we know that we've successfully recruited activity in that circuit. And then we can ask this really straightforward question about how other cell types respond to changes in neuronal activity. And we did this in the context of a diffusely infiltrating pediatric cortical glioblastoma from one of my adolescent patients. And what we found is that when we stimulated these cortical projection neurons in the premotor planning circuit, that there was circuit-specific increases in glioma cell proliferation. So just like their normal counterparts, OPCs, these tumor cells increase their rate of proliferation. And that this results in an overall increase in tumor burden, specifically within the stimulated circuit. So this was the first demonstration that brain activity can influence brain cancer growth. More recently, and in collaboration with David Gutman's lab at Washington University, we leveraged a genetically accurate mouse model of NF1-associated optic pathway glioma that the Gutman lab has spent many years developing and optimizing. In this mouse model, the tumors in the optic nerve form very consistently at precisely 9 weeks of age. And we can ask questions about the activity of the optic nerve and how that might influence the tumors that ultimately form there. So again, using optogenetics, we found that if we stimulated the optic nerve from 6 weeks onwards and then looked at the optic nerves at 16 weeks, that the tumors that formed in the stimulated mice were much larger than those that occurred in identically manipulated littermate controls in whom we simply did not turn on the light. Now, this optic pathway glioma model is a really exciting opportunity to ask questions not only about tumor growth, but also about tumor initiation, because again, we know exactly where and when these tumors are going to form in the optic nerve, and we don't really need to use optogenetics. It's the visual system. We can just change visual experience to modulate activity in the nerve. And so if we put mice into dark rearing conditions, we thought maybe in the beginning we could just put cool glasses on them. That doesn't work. I'm kidding. But if we put them into 24 hours of darkness around the time that these tumors form, or even just afterwards, so putting them into dark rearing conditions at 9 weeks of age or at 12 weeks of age, and then examining the optic nerves at 16 weeks of age. When we examine the optic nerves, we find that mice that were dark reared to decrease visual circuit activity had far fewer and much smaller tumors compared to littermate controls that were simply raised with normal visual experience, with normal 12-hour light and 12-hour dark cycles. And when we did a control experiment to say, well, is it that we messed up their circadian rhythms? We could control for that by light entrainment, 15 minutes of light at their waking and 15 minutes of light at their transition to night periods of 7 a.m. and 7 p.m. to keep their circadian rhythms synchronized. We saw no difference in this effect. We still did not see tumors in the absence of normal visual experience. If instead we put mice into complete darkness at 12 weeks of age, prior to the initiation of this tumor, that when we examine the mice at 16 weeks, there are absolutely no tumors compared to 100% tumor initiation in littermate control mice that were raised with normal visual experience. So there's some really powerful interactions between neurons and nerves and brain cancers. And over several years, we discovered that one major category of mechanism is that there are neuronal activity regulated secreted factors, paracrine factors that signal between active neurons or other cell types in the environment that are responding to activity and the cancer cells themselves. And those include some expected paracrine factors like BDNF, brain-derived neurotrophic factor, is something that we found to be a key paracrine factor mediating normal neuron OPC interactions. We've also found a shed form of a synaptic adhesion molecule called neuroligin 3. This is a very crucial signaling molecule between neurons and glioma cells. We find that it's also an important signaling molecule in myelin biology. Now, this neuroligin 3 discovery was kind of surprising. At the time, we didn't know that neuroligin 3 was any kind of mitogen in any kind of context, and we didn't even know it was cleaved and released. So we wanted to understand more about that. And in studying neuroligin 3, we wanted to understand how relatively important it might be. So there are many mechanisms that are intrinsic to the cancer cell that promotes growth and survival. There are several microenvironmental mechanisms at play. We wanted to understand the relative importance of neuroligin 3. So we simply, and this is work that was led by Hamsa Venkatesh, who's now in your town, on the other side of town in the Longwood campus, running her own independent lab. And what Hamsa did is she took these patient-derived, brain cancer cells and xenografted them into the environment of either the neuroligin 3 wild type or neuroligin 3 knockout brain. And what we found was absolutely unexpected and kind of stunning to us. In the context of the neuroligin 3 wild type brain, in a normal mouse brain, these GFP-labeled patient-derived, in this case, glioblastoma cells, they expand, they, infiltrate the cortex, they invade across the corpus callosum, they act the way GBM cells tend to act in both patients and in mice. But in the absence of neuroligin 3, those very same cells just stagnate. They persist. These cells were xenografted 6 months prior, but they don't expand. And you can measure that in real time using in vivo bioluminescent imaging. And you can see here that this pediatric glioblastoma model just did not expand over the course of the experiment. And this kind of surprising and apparent dependency of these gliomas on microenvironmental neuroligin 3 was conserved across multiple different glioma types, including DIPG and adult IDH wild type GBM. We also found that this was a key mechanism in that optic pathway glioma model, but we did not see this surprising dependency extend to a patient-derived model of breast cancer brain metastasis, suggesting that while neuroligin 3 is a really important mechanism across multiple different glioma types, it may not be conserved across all cancers growing in the brain. So why is neuroligin 3, this shed postsynaptic adhesion molecule, having such a profound effect on glioma growth? Well, in trying to understand the processing of neuroligin 3 and its downstream signaling pathways, we found that neuroligin 3 is shed in a strictly activity-dependent way through the enzymatic activity of a metalloprotease called ADAM10. It then binds on the glioma cell to a proteoglycan called CSPG4, also called NG2. Similarly binds to normal OPCs through the same mechanism. And then this recruits numerous oncogenic signaling pathways. There's early and upstream stimulation of focal adhesion kinase, downstream SRC, RAS, and M3 kinase, PI3 kinase, mTOR pathways. And that helps us understand the sufficiency of neuroligin 3 in promoting the proliferation of glioma, but it doesn't at all explain this unexpected dependency. And so we then dug a little deeper and looked at the gene expression changes attributable to neuroligin 3 binding and found that there were a number of synapse-related genes regulated by neuroligin 3. Looking at the whole cohort of really interesting gene sets that are regulated by neuroligin 3, we noticed that actually neuroligin 3 is regulating a number of different processes, including many ways that we now know neurons and cancer cells are interacting. But we really focused at the time on these synapse-associated genes. And you'll see that there's a feed-forward effect of neuroligin 3 binding on its own expression. So the cancer cells start to express neuroligin 3 once they're exposed to neuroligin 3. There's upregulation of the gene encoding the receptor for BDNF, TREC-B or NTREC2. But in addition to that, there are a number of AMPA receptor subunits and other synapse-associated proteins that are being regulated by neuroligin 3. And that raised, kind of a lot of questions in our mind. Number one, we wanted to validate that this was really happening in patient samples. So we collaborated with Mario Suba's group across town and found that in single cell data sets of each major form of glioma, including H3K27M mutant diffuse midline gliomas, IDH wild type glioblastoma, and IDH mutant glioblastoma, that in the malignant cells, there's robust expression of AMPA receptors, of neuroligin 3, of PSD95, and other structural and neurotransmitter related proteins. If we look at that data from kind of a cellular architecture standpoint and think about the cellular hierarchy in these tumors, here you're seeing data from the H3K27M data set. You can see that the cancer stem cell-like cell, which is in the case of DMGs is an OPC-like cell, is strongly enriched in these the synaptic gene expression. And actually, that's really interesting because it turns out that normal OPCs communicate with neurons not only through these activity-regulated paracrine factors, but also through functional neuron to OPC synapses. And so then I started to wonder if there are neuron to glial synapses in the healthy nervous system, might there also be neuron synapses forming on these malignant cells that are very OPC-like? And when we look, although it seemed like a crazy idea, when we look by immunoelectron microscopy, we see very clearly these synaptic structures between presynaptic neurons, and you can see these synaptic vesicles in the synaptic bouton, a clear synaptic cleft, and then this postsynaptic density on a cell that is immunogold labeled, that is a malignant glioma cell. Testing the idea that neuroligin 3 may be functioning perhaps in its most fundamental way to regulate these neuron to cancer synapses, we find that far fewer of these structural synapses form in the absence of neuroligin 3 from the tumor microenvironment. But are these neuron to glioma synapses just a shadow of the cell type from which we believe that these tumors emerge, or may they actually be electrophysiologically functional? So to test that, we performed whole cell patch clamp electrophysiology of the tumor cells, these GFP-labeled tumor cells that had been xenografted to a well-worked out and very experimentally tractable circuit in the hippocampus. By the way, DMG very frequently spreads to the hippocampus, so this is also kind of a clinically relevant paradigm. When we patch clamp these cells in the CA1 region of the hippocampus, we can at the same time electrically stimulate the axonal afferents into this region, the Schaefer collateral afferents. And when we do that, we see very clear postsynaptic currents in the cancer cell. These are dependent upon action potentials. They're blocked by the voltage-gated sodium channel blocker tetrodotoxin. They exhibit multiple electrophysiological characteristics of bona fide synapses, including paired pulse facilitation and single vesicle events called mini EPSCs. And this first kind of synapse that we identified are mediated by calcium permeable AMPA receptors. Now, normal AMPA receptor mediated synapses in the healthy brain and in between neurons are regulated by activity. Their strength can be changed in response to neuronal activity. They exhibit what we call synaptic plasticity. And one mechanism by which activity of a circuit can regulate the strength of a synapse in that circuit is through neurotrophin signaling, through BDNF signaling. And looking at the gene expression profiles of these tumors, we thought that perhaps BDNF to TrkB signaling was one way that synaptic plasticity might be at play. So Katie Taylor, when she was a postdoc in my lab, she's now leading her own lab at Memorial Sloan Kettering. When she was in my lab, she thought, well, let's see if there's synaptic plasticity at these malignant synapses. And we find that when we repeat that experiment, patching onto a malignant cell in a xenografted slice, and then perfuse BDNF over the slice, there are clear increases in the amplitude of those malignant synaptic currents. This depends upon expression of the BDNF receptor TrkB. If we CRISPR delete the gene encoding TrkB, encoded by NTrk2, then BDNF has no effect on the synaptic current. And what we determined was happening is that neuronal activity, which results in the release of BDNF, increases the trafficking of AMPA receptors to the postsynaptic membrane in the cancer cell. So the cancer cells are hijacking this classical mechanism of postsynaptic plasticity to increase the magnitude of the malignant currents. We wondered if there were other kinds of synapses. Glutamatergic neurons are the most common neuron subtype in the brain, second only to, secondarily are GABAergic neurons. And we wondered whether GABAergic neurons and especially GABAergic interneurons might be promoting the growth of some kinds of cancers. And when we looked at GABA-related gene expression, we found really interestingly that there was robust GABA-A receptor subunit and other GABA-A associated genes expressed in diffuse midline gliomas, in H3K27M mutant diffuse midline gliomas, much less so in IDH wild-type glioblastoma. And so we wondered whether there may also, the AMPA receptor dependent synapses, I should say, were present in DMGs, present in pediatric glioblastoma, present in adult glioblastoma, replicated at this, simultaneously in Frank Winkler's lab, who found the exact same thing. These GABAergic synapses we thought might be more tumor specific. And if we look at the, DMG cellular hierarchy, then we find that, just like synaptic genes in general, the GABAergic genes are somewhat enriched in this OPC-like compartment. So when we look for GABAergic synapses, again, using immunoelectron microscopy, and we did this in this case by fusing a GFP to a GABA-A subunit. So then all the immunogold labeling in the tumor cells are, indicating a GABA receptor. We see very clear neuron to glioma GABAergic synapses. You can see this through high resolution microscopy as well. Here we've cultured patient-derived DMG cells that we've engineered to express a GFP tagged GABA-A subunit, GABAG2. And then we co-culture them with these red neurons, and I'm showing you here neurofilament stained in red, together with immunolabeling of a presynaptic marker called synapsin-1. And you can see in this 3D reconstruction, very clear co-localization of the glioma-expressed GFP tagged GABA-A subunit and the presynaptic neuron-expressed synapsin-1. So structural GABAergic synapses are forming between GABAergic neurons and DMG. And we wanted to know if those were electrophysiologically functional. So, they are. We see very clear GABAergic currents in the tumor cells. These are GABA-A dependent. They're blocked by picrotoxin. They're also blocked by a different GABA-A receptor inhibitor called bicuculline. And interestingly, we find that these GABAergic synapses in DMG are depolarizing. Now, we think about GABA as an inhibitory neurotransmitter, and it is for mature neurons, but whether GABA causes hyperpolarization and inhibition or causes depolarization or excitation is just really dependent upon the intracellular chloride concentration of that cell. GABA is just a simple chloride channel. And so depending on the intracellular chloride concentration, chloride ions will either flow in or out of the cell, depending on, the gradient there. So we find that diffuse midline gliomas exhibit this depolarizing current in response to GABA. And I just want to point out here that IDH wild type glioblastoma, shown in blue, has essentially no current or a very, very small current in response to GABA. So again, this kind of tumor specific difference in the electrophysiology of H3K27M mutant DMG and IDH wild type high-grade glioma. Importantly, positive allosteric modulators of GABA-A receptors like benzodiazepines strongly augment the strength of these GABAergic currents. So we wanted to know if a stronger current might differentially affect the tumors. And what we find, and I think this is a really important clinical point, is that in H3K27M mutant DMG, quite specifically, we don't see this in GBM, there is a benzodiazepine-induced increase in tumor cell proliferation rate in vivo when mice are administered, in this case, lorazepam or Ativan. That results in an increase in overall tumor growth and a decrease in mouse survival. So just as we need to understand the neuroscience of these tumors so that we can identify medicines we may be able to repurpose to disrupt neuron-glioma interactions, we also have to be really mindful of the tumor-specific neuroscience of each of the cancers we treat and avoid medications that might be inadvertently harmful. It stands to reason that if GABAergic signaling is, depolarizing and promoting the proliferation of these tumors, that GABAergic neurons are also promoting the growth of DMG. So we tested that again using in vivo optogenetics. And we see that when we optogenetically stimulate GABAergic interneurons in the tumor microenvironment, this increases the proliferation rate of the cancer cells. So I've told you now that, both glutamatergic and GABAergic neurons can influence gliomas. I'm a pediatric neuro-oncologist focused on DIPG, so I study diffuse midline glioma. And in that disease context, both neuron types are promoting growth. For adult GBM, glutamatergic neurons play a larger role between these two. We've now looked at many other neuron types, again, especially the diffuse midline gliomas occur in midline structures. And we find actually that every neuron type that we've studied robustly promotes the growth of these cancers, cholinergic neurons, serotonergic neurons, noradrenergic neurons. So there's a lot of neurons influencing glioma. Does that mean that there's some convergent mechanism? Is there some way that all of these neuronal inputs are ultimately affecting the growth and progression of the cancer? Well, all of these different ways for the tumor cell in a neuronal activity dependent way to depolarize is a super metabolically expensive thing to do. And that suggests that, maybe this is the convergent mechanism. Maybe membrane depolarization alone is promoting glioma growth and progression through voltage sensitive signaling molecules or voltage sensitive signaling pathways. That would make some sense when we consider that actually in the developing nervous system, in the prenatal brain, neural stem cell niches exhibit synchronized waves of membrane depolarization and consequent calcium transients throughout the germinal zones. And that seems to be important for normal corticogenesis. So we tested whether membrane depolarization alone was sufficient to promote glioma growth. And we did that again using optogenetics, but this time we express these light-sensitive opsons not in the neurons, but rather in the tumor cells. And in this way, we can use light to optogenetically depolarize the cancer cell. And we can do that in an in vivo setting. So if we depolarize a xenograft, a glioma xenograft that is alone sufficient to promote cancer cell proliferation. Now, we're not just seeing these gliomas, undergo membrane depolarization, right? But they're recruiting mechanisms of adaptive plasticity to increase the magnitude of the depolarizing current. So maybe the more depolarizing it is, the more growth promoting it is. So we can test that again using the same system, but just flashing the light, to open the light sensitive, in this case, channel rhodopsin 2 cation channel for different time periods, which can cause either a medium depolarization of the cancer cell or a larger depolarization of the cancer cell. And when we do that, we see that there is a depolarization magnitude dependent effect on cancer cell proliferation in vivo. We can visualize this electrical activity in the tumors using genetically encoded calcium indicators like GCAMP6S. So this is GCAMP6S expressed in DIPG cells in vivo. And you can see really clearly what has been, for me, a very startling new understanding that these cancers are electrically active tissues. And that's not the way that we have been approaching either understanding them or treating them. And so now as a field, we really need to understand the mechanisms of malignant circuit assembly, plasticity, and evolution over the disease course, to discern the granular details of these voltage-sensitive mechanisms of cancer cell proliferation. This will give us both insights for patients' therapy, as well as insights for normal neural development and plasticity viewed through the magnifying lens of these glial cancers. What I'm telling you in summary is that GBM, DIPG, gliomas in general integrate into the circuits that they invade. They do this through bona fide neuron to glioma synapses that are then elaborated and amplified through mechanisms, hijack mechanisms of adaptive plasticity. They also do this purely electrically through activity-dependent potassium-evoked currents that are then amplified in a tumor-to-tumor cell gap junction coupled network. We know that this happens not only in mice, but also in patients. In collaboration with Sean Hervey-Jumper at UCSF, he measured using intraoperative electrocorticography and MEG imaging the functional connectivity between adult glioblastoma, IDH wild-type glioblastoma, and the rest of the brain. And the degree to which the tumor is functionally connected to the rest of the brain is a robust predictor of overall outcome, with more highly functionally connected tumors portending a much worse prognosis. We can begin to think about how we can target this therapeutically, disrupting neuron cancer interactions. Every way we do it in the lab slows tumor growth and progression. So we can think about, neuroscience-inspired targets that include neurotransmitter receptors, neurotrophin receptors, synaptogenic factor signaling, axon pathfinding molecules, synaptic adhesion molecules, ion channels, and gap junctions. There are excitingly clinical trials that are now beginning or have begun, including one that is collaborative between Stanford, MGH, and Dana-Farber. There's great therapeutic potential of disrupting these neuron-glioma interactions. As one example, if we disrupt AMPA-dependent signaling, either pharmacologically or in this case genetically, by expressing a dominant negative version of the AMPA receptor GluA2 in the glioma cells, this has a stark growth inhibitory effect on the tumor. So there are very clear mechanistic parallels between normal neuron to OPC signaling and malignant neuron to glioma signaling. So much so that it really underscores the extent to which these cancers are simply hijacking mechanisms of normal neural development and plasticity, and demand that we begin to approach these cancers from a neuroscience perspective. I want to introduce you to, or make sure I highlight the people who have been involved in the work that I've just shown you: Hamza Venkatesh, Ewan Pan, Katie Taylor, Tara Barron, Richard Drexler, and Avishai Gavish. Many, many people to thank, past and present, collaborators here and many in Boston, and of course, patients and families whose donation of tumor tissue enables all of the work that we do. Thank you so much for your attention, and hopefully we have some time now for questions.
Thank you, Michelle. I'm sure there will be a lot of questions for your exciting presentation. Jan.
Michelle, great talk. Thanks so much. I'm curious how, in the light of your work, how do you think about tumor treating fields and the mechanism of how those might be providing some benefit?
Yeah, I would love to know how tumor treating fields influences the activity of the neuron and the, depolarization of the glioma cells. I think I don't know the answer, but it does bring up the wider concept that neuromodulatory devices and approaches to perhaps drive a depolarization block or modulate this malignant circuit activity is something we should all be thinking about very hard.
Thank you, Michelle. That's really super exciting. I'm wondering, so obviously if you have a brain met from a different histology that takes place, takes root in the brain, right? Obviously the synapses won't be exactly the same because it's not a brain cell, it's like whatever, a breast cancer cell or lung cancer cell. But do you have any hunch or evidence, or is there any data out there that maybe the surrounding neurons around the met then would also have similar upregulation and activity that could then influence the growth of the met?
Yeah, actually, we've studied this. So this is collaborative work. Hamza is now leading her own independent laboratory at Dana-Farber, work she began before she left my lab, and that we've continued together collaboratively together with Julian Sage, and work led by, I just have to say, an absolutely brilliant medical student who just matched at Brigham and Women's Hospital. So well done. You get Salsa Savchuk, who's completely brilliant and led all of this work. We find that not only does neuronal activity drive small cell lung cancer brain metastases, but there are synapses. Now, this is a neuroendocrine tumor, so maybe that's not so surprising. But when these cells get to the brain, they make both glutamatergic and GABAergic synapses with neurons, and that very robustly drives their growth through what appears to be very similar mechanisms. Do
you think that the pharmacological inhibition of what you're talking about with GBM cells would then also apply to brain mets?
Yes, I do. Yes, I do. And Frank Winkler's group has shown, we both have these they're preprints that are up on bioRxiv, so you're welcome to read them. Frank Winkler's group has a study showing very clear EM evidence of synapses with breast cancer, with melanoma, and I think with non-small cell lung cancer. So I do think when tumors go to the brain, they adapt to take advantage of the currency of the realm, if you will.
I'm so sorry. Then I have another follow-up question. So,
sure.
The data that you show about lorazepam,
that
would then apply to mets from other indications as well, right?
So the GABAergic synapses that we observed, as I mentioned, were present in DMG, not present in GBM. We do see GABAergic synapses with small cell lung cancer, and those experiments with lorazepam are ongoing right now. I think it's important to identify, which tumors are safe to, be exposed to which symptomatic medicines. And we don't know, but we have found, some of the other strategies to disrupt those interactions that work in GBM also work in small cell lung cancer.
And then, of course, I'd be super curious what happens in melanoma brain mets and breast cancer brain mets.
Yes, Frank Winkler's group has this preprint out that shows synapses in both of those disease types. And in breast cancer, I should say, in addition to direct neuron to malignant cell synapses, there are also malignant cells that are sort of more astrocyte-like when they get to the brain and form perisynaptic contexts, a sort of pseudo-tripartite synapse that was described by Doug Hanahan's group. And that paper is published from 2019. In that case, glutamatergic signaling through NMDA receptors on the breast cancer cells drive breast cancer brain metastasis growth.
That was an awesome talk, Michelle, and thanks for joining us today. So I had a quick question about sort of trying to treat the tumor and the tumor neuron interface without affecting normal neurological function, because many of the glutamatergic receptors that that we might want to inhibit here are also present and important for normal brain function. How do you think about
and actually
that?
We can take some key lessons away from anti-epileptic medicine use. It's surprising that people tolerate those medicines as well as they do, but they do tolerate them well. They're designed to get into the brain. And when we repurpose medicines, for example, like the AMPA receptor targeting perampanel, we see that that slows glioma growth pretty robustly. And there are clinical trials that have already started using perampanel together with standard GBM therapy to see if that can augment the therapeutic outcomes.
Okay, we have one question from an anonymous attendee online, and they say, what are your thoughts on synaptic upscaling potentially playing a role in glioma progression?
Synaptic upscaling. Just to be clear, can the person who asked that question clarify a little bit what they mean by that? One thing that I will, while perhaps we wait for that answer, one thing that I'll mention is that there is, really, really clear effects not only of neurons and glioma cells, but conversely, and I need an hour-long talk to talk about this too, but there's conversely effects of glioma on neurons. Harold Sontheimer originally showed this. Ben Deneen has done beautiful work following this up. We have found, the same thing happening in DMG. There is a profound effect of gliomas on the excitability and structural and functional circuit dynamics of normal neurons and neural circuits. And that contributes to tumor-associated seizures, but it also augments the cycle, the sort of vicious cycle by which neurons drive the tumor cells increase the excitability of neurons. They do that through secreted factors that include synaptogenic factors, glypicans, thrombospondins, that increase the excitability and change the connectivity of neural circuits that increases the input to the tumor.
Right. They haven't posted anything after, but thank you for that answer.
Thank you, Michelle. I think I can ask you the last question. So you have really focused on the growth of the tumor cells as your readout for the excitability coming from the neuron. In some work we did with Frank Winkler and Varun Venkataramani, we also showed that there is an effect on invasion
Yes.
of the tumor cell.
Absolutely.
Do you think that this phenotype is also mediated by the same factors that you showed, neuroligin 3 or BDNF, TrkB, or do you think there is more to discover there?
Yeah, so two points. I think there's, just like growth can be stimulated both by synaptic mechanisms and by paracrine signaling mechanisms, we found that there's invasion, encouraged by neurons through both kinds of mechanisms. So the 2022 Cell paper that Varun led, beautiful work showing that the glutamatergic neurons synapsing onto the tumor cells can promote invasion. We've also found that there are secreted factors present in conditioned medium from, cortical slices regulated by neuronal activity that promote invasion. We published that together with Ben Deneen, and we spent a long time trying to identify the factors. It's not BDNF and neuroligin 3. There are unique factors that are promoting invasion of glioma cells that are secreted. We actually haven't identified them yet, but we know that there are both paracrine and synaptic mechanisms, and it's certainly a very important part of the effects of neurons on the tumor. And it's probably why, I don't know if the other clinicians in the room have also seen this, but at least in DMG, I tend to see cancers invade along circuits, like not in a just extension kind of manner, but for example, throughout, limbic circuitry, including crossing the midline and following the circuits to the next synaptic partner. I think activity clearly is influencing invasion, and activity is also probably influencing treatment efficacy. I think it's promoting survival in the tumor cells. That's less well studied. But Frank's work shows, at least through the gap junctions between the tumor cells, that there's treatment resistance.
Thank you, Michelle, for the great presentation. I think we can stop here. Thanks a lot.
Sure.